Hi, in this video we're going to talk about how eukaryotes regulate their gene expression. Now, necessarily this is going to be more complex than how bacteria regulate their gene expression because, well, eukaryotes have more complex genes and more complex chromosomes. So, you actually will see regulation in eukaryotes at the structural level of the chromosome. So, the chromosome, as we know, is made up of a strand of DNA. Here we see that DNA strand right here. And it's wound around these things called nucleosomes. So there we see a nucleosome, and here is the DNA wound around it. And if we zoom out a little bit, looking at this next picture, we see that this forms a pattern that's usually referred to beads on a string, because these little clusters with the nucleosomes kind of look like beads and the rest just looks like a string. Not important that you remember that name, it's more just an analogy of this structure. So here we have the beads on a string structure. And this can actually be clustered very tightly together, like we see in the next image. It can also remain in this open state. So these two images that I've drawn the double arrow between, those are not mutually exclusive. They exist together in different parts of the chromosome. And we'll talk about why some are one way and why others are the other. So lastly, if we zoom out all the way, we kind of see this structure here, just this condensed chain of DNA. However, as I mentioned, some parts remain open in that beads on a string shape. And the reason is, those are the parts that contain genes that need to be transcribed. So in eukaryotes will open up their chromosomes in the areas where gene transcription is needed, and they'll condense their chromosomes in the areas where gene transcription is not actively needed. So one of the ways they do this is with histone acetylation. So those nucleosomes that we just saw, those are actually made up of 8 histone proteins. So here in this model you can see that these discs, they've been cut into quarters, and you may or may not be able to see it, but they've actually been cut along the side too to sort of represent 8 pieces, little eighths of this nucleosome. Each one of those is a histone, and each histone is its own protein. And what can happen is you can acetylate these histones. You can add an acetyl group like we see here, and when you acetylate the histones in these nucleosomes, it opens up the chromatin structure. So here we have what is called relaxed chromatin, and here we have the closed chromatin. So whether or not our histones are acetylated, we will either open or close our chromatin, allowing it to be easily read or making it impossible for the machinery to come in and transcribe various genes. Now, another way that we can regulate gene expression at the structural level of DNA is with DNA methylation, which is the addition of methyl groups to adenines and cytosines in DNA. And this essentially serves to regulate gene expression, much like we saw with the histone acetylation, deacetylation. Methyl groups are kind of bulky, so when attached to these nucleotides, they make it difficult for the machinery of DNA to come in and read those base pairs. So by methylating DNA, we make it more or less unreadable to the genetic machinery. And that's just another way that eukaryotes regulate gene expression on the structural level. Now let's turn the page and look at some other ways that eukaryotes regulate their gene expression.